Hi, my name is Adriana Villacis, and this is my senior project presentation. My central question is, what is the impact of precipitation on E. coli in the Charles River watershed? So the outline for this project, I'll talk about my inspiration, my process, my data, and some challenges that I faced throughout this project. So why I chose this topic, I've always had a passion for biology and environmental science, and I plan to continue that through college. It also allowed me to look into a potential career of data science, and I also just wanted to learn more about public health in our local communities. Vision of a graduate, so I developed my communication skills immensely by talking to people from outside organizations, contacting them through email, Zoom calls, and a lot of more ways. I also developed my critical thinking while I was constructing my graphs, and that definitely took a lot of time and challenges, so I had to work through those. And lastly, collaboration throughout my project, because I wasn't solely focused on people from Villacis High School and was able to communicate with people from the Watershed Association. So my timeline, I started this project in the summer of my junior year, where I became a volunteer for the Charles River. I then collected precipitation and bacteria data throughout October and December. After that, I created graphs for each lab site in Boston, which there are 6 of them, and I'll go through the data later in this. I also then presented to the Environmental Action Club in March and created pamphlets about the prevention of E. coli and then distributed those pamphlets. So my mentor, Maria Elena Lima, she is a data scientist for the Charles River Watershed Association. She is also the River Science Program Manager, so our work relationship, we met bi-weekly. I initially contacted her through the CRWA website. She also assisted me while creating my graphs and gave me a lot of feedback during my major tasks that I had to complete. So a little bit of background before I jump into my project. E. coli is basically fecal matter that is usually found in bodies of water that will contaminate it. So the way that E. coli gets into these bodies of water is that it'll be on impervious surfaces. So for example, an impervious surface would be like a flat driveway, and one that isn't is just like a rigid driveway that isn't paved. So that way when there's excess precipitation, the runoff will go into the street, as you can see in the photo, and that'll go into the sewage systems where there's like a lack of filtration, and then eventually go into the bodies of water and contaminate them. So I started off as a volunteer monthly monitor. So as you can see, this is kind of like the lab site. It's on one of the routes in Villacis. So I trained with my volunteer coordinator. He taught me how to complete the entire process. I also contacted my sampling partner, which her name is Kimberly Schubert. So we kind of worked hand in hand every third Tuesday of the month at 6 a.m. to complete this task. It took about 40 minutes to an hour. And these samples were then sent to lab sites to look for different bacterias. So my sampling process, two sample bottles were lowered into the river. As you saw on the slide before, we recorded the temperature of the water in degrees Celsius. Then we measured the depth of the river with a rope with a weight attached to the bottom of it. We then recorded the data on the collection paper, as you can see. So the actual location is Route 115 on Baltimore Street in Villacis. It's like that little bridge, if you know where it is. We kind of just write our observations in there, and then we send those samples to lab site. So this is my data for Watertown and Boston, both sites, just different locations. So I collected data for 3 years, so 2021 to 2023, and kind of put them all in this graph. So as you can see, E. coli is on this side and precipitation at the bottom. And there isn't much of a correlation, but throughout the graphs, you can kind of see that there's slightly a positive trend. So these are also 2 Cambridge sites in Boston. As you can see, there's a lot of outliers, a lot of mixed data. And lastly, these are the last 2 sites, another area in Cambridge and one in the Charles River Dam in Boston. And I'll talk about all my results next. So overall, for my results, I believe it's inconclusive. As I said, there's a slight positive trend within some of the graphs, but mostly outliers. And that's basically due to wet and dry seasons for the past 3 years. And I talked to my mentor about that, and we kind of analyzed all of my data together, and we came to that conclusion. And overall, there's minimal amounts of E. coli in the actual river, so if you were to go swimming in it, you won't actually get any or contract any diseases that are fatal. So next steps, after analyzing my data, I knew that sampling more than once a month was definitely necessary for this project, or just collecting more years of data instead of 3 years, maybe 10 years. So then I knew with that I wanted to present to those who are willing to continue this project now that I had an idea for a continuation, and I also wanted to create brochures to spread awareness on this topic. So the first way I spread awareness was I presented to the Environmental Action Club, and I thought this was a great opportunity because they always focus every month on a specific topic, especially like climate change and other bacteria such as what I'm studying right now. I explained the volunteer opportunities on the website that I initially used in the summer. I gave an overview of my project and explained all my data and my results, and explained what I thought could be a good continuation of it. So spreading awareness, I created the pamphlets in front of you if you want to take a look at them. So I created these on Canva, and I used the research throughout the year to make them. I printed and folded each one and distributed them. So I'll give you a little bit of an overview of what I wrote. So on the first page, it talks about what E. coli is, just to make it, just to have a baseline. And then I talk about the causes and effects of stormwater sewage. And in the middle, I have even a photo of my project and all the data that I found and kind of analyze it a little bit. I then talk about what runoff is and how to prevent it. And the tax, I'm sure that you guys know that there's a property tax on impervious surfaces and how you will be taxed that. So it kind of explains what that tax goes to and that it's not going anywhere, that it's actually important. And a little bit about who I am and my contact information in case anyone wanted to reach out and ask me more questions. So these are the distributed pamphlets. I'm not sure if you saw it today on your way in. So I thought the Millis Public Library would be a great way to spread the word because I know there's a lot of advertisements there already and that many people walk in and out of all ages, so I thought it'd be good. And the Millis Town Hall was my second area that I decided to display my pamphlets because I know there's many people with bigger voices than I have that walk into the town hall and that are able to speak at town meetings and possibly introduce this topic during that. So some challenges that I faced was minimizing my data to what was necessary. So throughout this project, when I initially gathered all my data, it came with the depth of the river, the temperature, all of that. So I had to cut down and really play with a Google Sheet to make it specific to each site and everything, as you can see in the photo. So that was definitely a challenge. Also, constructing my graphs was especially difficult because I wasn't sure on what specific graph to do, but after researching, I came to the conclusion that a scatter plot would be the best for this. That way you can see the outliers and how E. coli definitely varies depending on temperature, precipitation, all of that. And lastly, a big challenge was that my hypothesis was refuted. Unfortunately, I didn't have any say over it, but overall, I think that if my project was to be continued again, that I think after collecting more years of data that it could possibly be supported in later years. And a big thank you to my mentor, Maria Elena Lima. I wouldn't have been able to do this without her. She taught me a lot about data science, E. coli, and everything. Also, Ms. Copese and Ms. Cheney for helping me with the ideas for my project and allowing me to think of new ideas and kind of just talking to them about it. And lastly, my VMM partner, Kimberly Schubert, because it was great to have someone there talking to you at 6 in the morning when you're taking river samples. And that's it. Thank you.
Good job, Adriana. I know that I learned a lot just reading your papers throughout the year about this because we put a deck on last year and we got charged for like the stormwater fee, and that was, I don't even know what that means, but now I do. I have 2 questions for you. I know you mentioned if you were to pass this on, like you would have them kind of go back a little bit further in data.
Yeah.
Do you think that this is something that someone should take over? Are you like, I mean, obviously one of your challenges was that your hypothesis was refuted.
Yeah.
But do you think that there's value in continuing it, or do you feel like you were kind of like grasping?
I think, at least for like the STEAM scholars, I think a lot of people struggle with finding like ideas that they can do.
Mm-hmm.
So I think it could be a possible continuation, especially if they did it that long, or if they wanted to, if they found like supportive evidence, they could do it at a town meeting, because that was like initially what I wanted to do. But after realizing my hypothesis was refuted, I wasn't able to do that because it just confused the public.
Right.
So I think that if it was supported and they did do it, that could be good too.
Nice job. I don't really have any questions, but I do want to say that you did a really nice job on this, because one of the things that you did was you picked this, this blue color, and it caught my attention. And, with the white on it, so it's just like a simple, like, graphic design thing that, as I was walking by, that was the one that I can't even tell you anything else that was there. So I did notice this one.
Thank you.
Yeah, on that note too, your slides looked really nice. They were really clean and they presented well. So yeah, you did a really nice job. You were really poised. My question was, how did you get involved with the Charles River Watershed Association as a volunteer?
Yeah, so initially, like, Miss Cheney kind of gave me somewhat of this idea, and then she told me that Miss Hickey had a contact at the Charles River. So I originally contacted her. I think her name was like Dyra Johnif. I don't know if I got that right. But after that, then she gave me another contact about the volunteer, and then after that, I finally found my contact, which was my mentor, Marilyn Alima. So I definitely worked through like a lot of people in that organization. So it was good just to like get my name out there too for future stuff.
Is this something you would pursue a career in data collection, or is this something that you're like, I'll do?
Yeah, after researching everything and constructing graphs, I don't know if I love data science just because it takes a really long time. And it's like a lot of numbers, and it kind of gave me like a headache while doing it. But I like more of like the sampling and all of that, so.